Hello, welcome back to our SC Pharma YouTube channel. Today we are going to discuss about the hanging drop motility test. The requirements for this motility test are: first we require the microscopic slide with depression, which is also called as cavity slide. Next we require the cover slip. Next is the paraffin wax or petroleum jelly. Next requirement is we require the young broth culture of the motile bacteria. Next one is the inoculating loop and the microscope. Procedure for this motility test: first step is to preparation of the cover slip by dabbing the petroleum jelly on the corners. That is, we are going to place the petroleum jelly on the corners of the cover slip. Next step, that is, place loop full of microbial suspension into the center of cover slip. Next step, we are going to put a loop full of microbial suspension into the center of this cover slip. Next step, turn the concave depression glass slide upside down over the cover slip. We are going to press the cavity slide upon this cover slip with gentle pressure. Next step will be the invert the concave depression glass slide to produce the hanging drop of this specimen. That is, we are going to invert this cavity slide upside down so that it will form a hanging drop on the slide. Here is also the same, that is procedure. We can also prepare the ring of Vaseline or paraffin wax as shown in this diagram. This is the cavity slide where we can prepare this ring of paraffin wax. Next, on the cover slip, we are going to take a drop of microbial culture. Then we are going to place the slide over the cover slip, and the last step will be invert the cavity slide upside down so to form a hanging drop over the slide. Here, this is the result of the bacterial motility seen under the microscope. Here I have attached the video showing the results of bacterial motility. You can see the video for the results. Here you can see the motility of the bacteria, how the bacteria is moving, which shows that the given suspension is a motile. Once again, look at this video where you can see that the bacteria are moving. So the conclusion will be the given bacterial suspension is motile. Thank you for watching this video. Please like, share and subscribe.